What is up? Hi, it's Coach Heichel. Today I'm bringing you a podcast over the basic structure of DNA, what DNA actually stands for, what it does, and some basic scientists on the discovery of DNA. So let's start with what actually it stands for. Well, the D in DNA is going to stand for deoxyribose. It's going to be the main sugar in the DNA molecule. The N and the A are going to stand for nucleic acids. If we remember from the first unit of the year, we know that nucleic acids are made up of repeating nucleotides. So a repeating nucleotide, one nucleotide is made up of three parts. It's made up of a sugar group, which is going to be your big pentose sugar right here in the middle, this big blue house-looking structure. That's going to be your deoxyribose in this case. You're also going to have a phosphate group. Phosphates will always be circular when you're looking at a 2D picture of them like this. So this is going to be your phosphate, and these right here in the middle are going to be your nitrogen bases. Your nitrogen bases is what makes DNA unique. There's four of them. There's going to be what's called adenine, represented by an A, thymine, which is going to be represented by a T, a cytosine, which is going to be represented by a C, and then you have guanine, which is represented by a G. So these four nucleotides, or nitrogen bases, make up every living thing on this earth, just in different combinations. So that takes us to how they actually pair together. So we know we have an A, a T, and a C and a G. So how do they pair together? Well, C is always going to be paired with G, or cytosine is going to be paired with guanine. They're going to be held together by three hydrogen bonds. Adenine and thymine are also always going to be paired together. So A to T, C to G. A and T, though, however, held together by two hydrogen bonds. So A is going to go to T, and C will always be paired with G. There's two kinds of bases that we need to be familiar with as well. We've got what's called pyrimidines, which are going to be your single ring bases, like this here. It's made up of only one ring of carbon and nitrogen atoms. And there's also your purines, which are going to be your double ring bases. Thymine and cytosine are going to be your pyrimidines. So again, pyrimidines, they have one ring of carbon and nitrogen atoms. Adenine and guanine, on the other side, are going to be your purines. They're made up of two rings of carbon and nitrogen bases. So there's the two rings. We got adenine and guanine. So purines and pyrimidines bond together. DNA is what's said to be double-stranded, two-stranded. It's kind of a double helix shape, but when we put it in a two-dimensional model, it kind of looks like a zipper. Okay, it's going to look something very, very similar to this. So again, these circles are going to be your phosphates. This right here is going to be your sugar, if we remember that. In this case, it's going to be deoxyribose. And these in the middle are going to be your nitrogen bases. So if this is an A, this one on this side is going to be a T. And we talked a few slides ago about how hydrogen bonds hold the bases together. So you can't really see the hydrogen bonds in these, but that's what's holding them together. That's what's causing them to stick together. So if you wanted to separate this molecule, all you'd have to do is find some kind of chemical that could break down hydrogen bonds. So just a few more tidbits on hydrogen bonds. Hydrogen bonds themselves aren't very strong. They're actually a very weak bond. But when you put in millions and millions of them together, it actually makes a pretty stinking strong DNA molecule. It's hard to break down. And here we just show the hydrogen bonds between guanine and cytosine. Okay? Remember, the purine is going to be your double ring. The pyrimidine is going to be your single ring. So again, when making hydrogen bonds, C always pairs to G, and A always pairs with T. The structure of DNA is actually pretty basic. It's going to have a 5' end and a 3' end. The 5' end will always be the phosphate, which is represented by this P. The hydroxyl end, or the 3' end, will always be an OH, which is called a hydroxyl group. So it's what's said to be anti-parallel, because on this side, we start with our phosphate and we run down to our hydroxyl. But on this side, we start with our phosphate and we run up to our hydroxyl. So we have a 5 here and a 3 here. That's what makes it anti-parallel. It's completely opposite, but it's parallel because these will never touch each other. And again, we have our nitrogen bases in the middle being held together by hydrogen bonds. It's important to note that these nitrogen bases are only attached to the sugar molecule. They're not attached to the phosphate. So it's a phosphate, sugar, so on and so forth, with nitrogen bases only attached to a sugar. The basic function of DNA is to store genetic information. So if you take everybody's DNA in this entire world, every living thing, plant, animal, down to the smallest little fish, jellyfish. Okay, they're all going to have the same 4 bases. They're all going to have an A, a T, a C, and a G. Okay, those are going to be the 4 basic nucleotides that every living thing is going to have. It's just the combinations of them that make them unique. So history and sciences. Basic history, we got a little bit more to go and then we'll be done for the day. Frederick Griffin, he was kind of the first to start tackling what DNA was and kind of gets credit for kind of discovering it, but not really, because he didn't really do a whole lot. He simply noticed that the instructions contained in the DNA genes were transmitted by chemical compounds. He did this in 1928. Didn't know what they were or anything like that. He just knew basic instructions were there. Rosalind, Franklin, and Maurice Wilkins. Now we're stepping it up to 1952, so it's still pretty new science. What they did is they took a DNA molecule out. They put it in what's called an X-ray crystallography. So they crystallized it, then they took an X-ray of the crystal. And they came up with this similar shape right here. Now this is looking down straight through the center of the molecule. So these are going to be your sugar phosphates all the way down the side. You can't see your A's and T's on the inside, your C's and your G's. They're just too small, you can't see them. A year later, the two guys that get the main credit for discovering DNA are called James Watson and Francis Crick. Now what they did is they used the picture that Franklin and Wilkins took, and they determined the structure. They said it was a double helix. It was kind of this spiraling staircase you see on the side of this PowerPoint. You know, it goes something kind of like this. And then it has your nitrogen bases are going to be connected in the middle, okay? And there'll be a connects model for you to see whenever you're going through this. So Erwin Chargaff, before Watson and Crick in 1950, came up with the determination that A equals the amount of T, and that C equals the amount of G, which is great because we know A and T always bond together, and we know that C and G always bond together. So he said that if you have 30% A, you're going to have 30% T. More on Erwin Chargaff, back to the 30-30 rule. So if you have 30% A, you're going to have 30% T. These two together equals 60% of the DNA molecule. So the other 40% has to be split up between this C and this G. So you take 40 and you divide it by 2. And you're going to get 20% is going to be C. You're going to get 20% is going to actually be G. So if you have 30% A, 30% T, 20 and 20, those two together equal 100%. Chargaff's rule is correct. And one last thing, just in closing, just to review the basic structures of everything we've talked about. So these circles, again, these circles are going to be your phosphates. These big things right here in the middle are going to be your sugars. The nitrogen bases are connected only to the sugars, and your sugars and your phosphates alternate as you go down the DNA molecule. So if this is a G, this molecule over here is going to become a C. If this is a C, this molecule right here, this nitrogen base, it's going to be its complement. It's going to become a G. So C, then we're going to have a G. Since we have a T here, what complements a T is an A. So we're going to have an A. Again, we have a T, so we're going to have an A. We have an A here now, so its complement is going to be a T. We have a C, so its complement is going to be a G. Got it? Great. If you have any questions, please feel free to email me. Again, this has been Coach Heichel. Until next time, we'll talk about DNA replication. Have a good day.